Hi everyone, welcome to my A-level biology lecture series. In this lesson, we will explore the structure and functions of mitochondria. At the end of this lesson, you will be able to describe the relationship between the structure and functions of mitochondria using diagram and electron micrograph. Let's get started. Mitochondria are present in nearly all eukaryotic cells, including those of plants, animal, fungi, and most unicellular eukaryotic cells. Mitochondria are double membrane-bound organelles, which means that each mitochondrion is surrounded by two layers of the phospholipids bilayer embedded with some specific proteins. The outer membrane is smooth. However, the inner membrane is highly folded. So the folding regions of this inner membrane is known as cristae, singular, crista, plural, cristae. So the inner membrane, divides the mitochondrion into two compartments, which are intermembrane space, a very narrow region between the inner and the outer membrane, as well as the mitochondrial matrix enclosed by the inner membrane. Let us look at this structure. So first of all, we can see that we have the outer membrane, which is smooth membrane, and inner membrane, which form cristae when we have the folding regions. So the space between the outer membranes and inner membrane is known as intermembrane space. So the folding of this inner membrane form crista. Okay, so singular crista, plural is known as cristae. So on this crista or cristae, okay, we will be able to find this structure known as a stop particle, or in other words, it actually consists of this ATP synthase. So ATP synthase catalyzes the formations of ATP. So the inner regions, okay, is known as mitochondrial matrix, enclosed by the mitochondrial inner membrane. So within this mitochondrial matrix, we will be able to find circular mitochondrial DNA, which is a double-stranded DNA, but it's in a circular form, as well as 70S ribosome. The mitochondrial matrix contains circular mitochondrial DNA and also 70S ribosome. So why? The question here is why there are mitochondrial DNA as well as 70S ribosome, because the gene in this circular mitochondrial DNA are used to code for a particular mitochondrial protein, and 70S ribosomes synthesize those proteins. For example, those enzymes used to catalyze the key stages of aerobic respiration, including the Link reaction, as well as the Krebs cycle. Right? So this 70S ribosome also synthesizes some these electron carriers, which are protein, and also the ATP synthase. So this electron carrier as well as ATP synthase are embedded in the inner membrane, which function in oxidative phosphorylation to produce or to synthesize ATP. So the table summarizes the features of each structure of a mitochondrion, as well as the specific functions of these structures. First of all, let us look at the outer membrane. So the outer membrane consists of phospholipid bilayer containing transport protein. And as we know that phospholipid bilayer is selectively permeable, and this selectively permeable membrane separates the internal mitochondrial environment from the external environment, the cytoplasm. And this external or this outer membrane is permeable to small molecules, for example, pyruvate, reduced NAD, as well as oxygen. So do not worry about those chemical or those compound. We will be learning them soon. So because this outer layer permeable to the small molecule, so this is very important. It's essential to allow those key molecule for aerobic respiration to enter into the mitochondria, particularly mitochondrial matrix. Next structure, let us look at this inner membrane. So against this inner membrane, they are highly folded into a state. So as we know that because highly folded, this increases the surface area for electron transport chains as well as the ATP synthase. So the larger the membrane, the more electron transport chain, the electron carrier as well as ATP synthase can be embedded on this inner membrane. Hence, it increases the rate of oxidative phosphorylation. So on this membrane, there are electron carrier, which facilitates the electron transport as well as to generate a steep proton gradient for oxidative phosphorylation. And as well as the electron carrier, we can actually find ATP synthase, the stop particle on the inner membrane, and the stop particle contain this ATP synthase, which catalyzes the formations of ATP during oxidative phosphorylation. And the inner membrane, one specific features of inner membrane is impermeable to protons. So therefore, we can actually maintain a very steep proton gradient, which is essential for chemiosmosis to occur. The next features or next structures, let us look at this intermembrane space. So intermembrane space or mitochondrial intermembrane space is a narrow compartment with a very low pH because it contains high proton concentration. So because of the narrow compartment, so it enables rapid build-up of the proton gradient for ATP synthesis. The next structure, let us look at the mitochondrial matrix. As we know that mitochondrial matrix contains mitochondrial circular DNA as well as 70S ribosome. So with these two components, mitochondrial circular DNA as well as 70S ribosome, so these features enable the synthesis of the mitochondrial protein and enzyme which involve in aerobic respiration. As well as this mitochondrial circular DNA and 70S ribosome, many enzymes that in charge for the link reactions and Krebs cycle can be found in mitochondria matrix. So those enzymes catalyzes the key stages of aerobic respiration to produce reduced NAD, reduce FAD. As we haven't gone through the entire process about aerobic respiration, you may find those terms, for example, link reaction, Krebs cycle, reduce NAD, reduce FAD, stop particle, ATP synthase, something quite very new to you, but do not worry at this moment. We will be going through the entire process of aerobic respiration. And when we have done this aerobic respiration, come back to this topic again or this lesson again, you will be able to get the overall idea and overall view about the structure and how this structure adapts for its functions in mitochondria. So with that, we have come to the end of this lesson. I hope you have found this lecture series helpful. If you have any question, feel free to leave them in the comment sections. See you in the next lesson. Have a great day.